Yes, that's right. And thank you so much for having me today. So my name's Dana Wyman. I'm from the Mortazavi lab at the University of California, Irvine, and my talk is going to be about some work we're doing as part of the ENCODE consortium to generate transcriptomes using long-read PacBio data. And specifically, the talk will be about some error correction methods that we're using. So first of all, short reads have been extensively used to quantify gene expression in a variety of systems, but there's been an increasing recognition that that doesn't give you the full picture. Specifically, we're interested in what's going on in the isoform level. And short reads are sort of challenging to work with for this purpose due to their short length, but long reads, such as PacBio, can be up to 60 kb in length, which allows them to cover entire human transcripts. And with that in mind, the goal of our project is to use long reads to obtain isoform level transcriptomes for different ENCODE cell lines and samples. But one trade-off of using long reads is that the error rate tends to be higher than for short reads. And one way that PacBio deals with this specifically is they use smart bell adapters that go onto the sides of the transcript molecules. And this allows the DNA polymerase to circulate around and around the transcript, producing multiple passes that can be compared to each other for computational error correction. And this works great most of the time, except when you get one or two passes, and this prevents the majority rules error correction from working, so you can get errors that do persist to the next stage. Specifically, Microindle errors can be a problem for PacBio. So these are small insertions or deletions of around 5 base pairs in size. And when these happen on a splice junction, you can get a problem when you map the reads to the reference genome, producing an artifact non-canonical splice junction. And because splice motifs are extremely conserved, these non-canonical motifs are considered artifacts. And up to 15% of the PacBio reads we see have at least one non-canonical splice junction. Unfortunately, the downstream pipelines usually discard these reads, so you're looking at losing a lot of your data to this problem. To deal with this, I wrote a program in Python called Transcript Clean, which corrects common sequencing errors in mapped long reads. So specifically, it will correct microindels, mismatches, as well as these non-canonical splice junctions that are close to an annotated junction. But I know that one size doesn't fit all, so I've created flexible input options so that you can turn off mismatch correction if you're concerned about RNA editing or SNPs. Or alternately, if you're working in a well-studied cell type, you can provide a whitelisted file of VCF variants that will prevent variants in those positions from being corrected. So an example of this can be seen on the left with a mismatch that matches a known SNP, and then we have a microindel in the middle there that gets corrected. And finally, a non-canonical splice junction on the right that's corrected. And this works on both PacBio and Oxford Nanopore data. So in our recent paper, we ran Transcript Clean on publicly available GM12878 data from Tilgner et al. 2014. And if you look at the table, you'll see that we corrected the vast majority of insertions, deletions, and mismatches. And we did use a VCF file specific for NA12878, so that's why you have variants there that were not corrected. Finally, we corrected 39% of the non-canonical splice junctions, which, if you look at the bar plot, amounted to a pretty nice reduction in the number of transcripts that contained one or more non-canonical splice junctions. Specifically, we rescued about 32,000 formerly non-canonical transcripts, which meant that they could be used in the downstream workflow to call isoforms. So we ran a downstream pipeline on these isoforms and on these transcripts as well as the other canonical transcripts to see the effect of running Transcript Clean. So we corrected errors, and then we collapsed the transcripts into isoforms. Then we removed isoforms with less than 2 supporting reads, and we annotated them to find out which ones belong to which genes and so on. And of the roughly 10,000 isoforms that we called in this pipeline, about 1,500 had support from one formerly non-canonical read that we were able to correct. And in addition, we had 600 isoforms that would not have been discovered at all in our data if we had not run Transcript Clean. So to look at a specific example of this, the 2 starred isoforms here for the gene ERI3 were specifically rescued by Transcript Clean. And so that can give you an idea of, compared to the rest of the data, how Transcript Clean helped us recover isoforms that we would not have seen otherwise. So in summary, Transcript Clean corrects errors in mapped long reads, which can help you increase the isoform yield downstream and make more use out of your long read data. And with that, I'd like to thank the Mortazavi lab, of which I'm part, as well as the Wold and Spitali labs. I'd like to thank our funding and the organizers for having me speak. And then if you're interested in learning more about this, you can get the software from my GitHub, DEWyman/transcript clean, and learn how to use it there. And you can also come see my poster or check out our paper in bioinformatics. Thank you very much.
How does the upstream software or Transcript Clean deal with real non-consensus splice sites?
Right.
GC and U12 being the two major cases.
Okay, so the question is how Transcript Clean would deal with actual non-canonical splice sites that are actually in the data. And specifically for the cell lines we're looking at, we have the advantage of having short read data, so we derived our splice junctions from mapped short reads to the genome. And so in theory, if the splice junctions are real, then they should be represented there, and you would allow the splice junction to stay if it matched that. Any other questions? Over there.
What would you say is a good depth of sequencing for Iso-seq data to get a decent coverage of relatively all the transcripts?
Okay, so the question is about coverage for Iso-seq data. And what we're finding, our sequencing facility recently upgraded to the SQL machine, and we're planning to go to 1 million raw reads per biological replicate. In the past, we've done at least 300,000 reads, but we don't think that's quite sufficient. So we're hoping 1 million will do it. Anyone else? Great, well, thank you.